This is the Professor, CBMD PhD, on today Sunday, March 23rd, 2025. It is 5:33 and 29 seconds, p.m. Eastern Standard Time, and today's research, DNA repair. So let's hop to it here. Even the most cooperative groups run into trouble when resources become scarce. Biofilms cannot grow forever. Eventually, new members of the group find there is a limit to how much ATP can be extracted from even the richest hydrothermal vent, and they may not survive when resources drop. This sets the stage for the next major step in the evolution of life on Earth: competition between teams of organisms. Fortunately, the same concept we discussed to explain how early protocells competed to survive applies here. Evolution by natural selection. To explain how this happened, we need to account for the same three traits: variation within the population, heritability of the variation, and the survival advantage for possessing this variation. The variation arises from two primary sources: built-in errors in DNA replication and exchange of DNA among members of a population. Although our definition of life requires an organism to replicate and self-repair, neither of these traits must be 100% accurate. A simple principle of physics is that disorder in any closed system, such as an organism and its energy source, always increases over time. In this context, it means cells never replicate their DNA perfectly or fix all their mistakes. Genetic replication errors are a fact of life. Because DNA stores genetic information, this variation is inherited by an organism's descendants. Prokaryotes even promote this genetic variation by sharing pieces of their DNA with their neighbors, which biologists call horizontal gene transfer. This accelerates the rate of evolutionary change by allowing members of a population to share the benefits of the variation without having to develop it themselves. An excellent example is antibiotic resistance. If a single bacteria mutates so that it is no longer susceptible to antibiotic drugs, it can quickly pass on that genetic information to its neighbors by copying the DNA encoding this resistance and physically transferring it to other cells it comes into contact with. This type of gene transfer can change prokaryotic populations dramatically, even giving rise to new species. The selective advantage of genetic variation within the population is, therefore, the determining factor as to how well organisms compete with one another. We can use biological codes to help explain this phenomenon. When genetic variation gives rise to a new or modified biological code, this can provide a tremendous advantage over organisms that lack it. One example is the code called quorum sensing. Quorum sensing is a bacterial language through which the microorganisms communicate with each other to regulate the size of their communities. Signaling molecules exchanged among cells can contribute to the formation of biofilms, colony diversification, prokaryotic motility, and virulence factor production. In some cases, this communal living is vital for the survival of an organism. Some prokaryotes can remain alive in their planktonic state, but can only replicate when they are in a symbiotic environment such as a biofilm. This is our first principle at work. Teamwork promotes prokaryotic evolution by increasing genetic diversity in the population. Eukaryotic cells comprise the third and most recent domain of biological organisms. When viewed under a microscope, the most striking feature of eukaryotic cells is that their cytoplasm is highly organized. Even the simplest microscope reveals the presence of a large, often oval-shaped structure called a nucleus. Closer examination with more powerful microscopes allows us to see additional distinct structures in the cytosol, such as mitochondria. These structures are generally classified into two groups. Cytoplasmic structures that are surrounded by at least one distinct membrane are called organelles. The presence of these membranes allows the cell to create specialized compartments in the cytoplasm that are devoted to performing a subset of cellular tasks under optimized conditions. Like the plasma membrane, these membranes are selectively permeable to ions and other small molecules, which helps to create a unique internal environment optimally suited to the molecules contained inside. Because it is a membrane bound, the nucleus is classified as an organelle. Additional organelles found in eukaryotic cells include the endoplasmic reticulum, Golgi apparatus, mitochondria, chloroplast, endosomes, lysosomes, and peroxisomes. Each of these organelles contains unique molecules that performs a separate set of functions. Large molecular complexes that are not enclosed in a separate membrane do not have a generic name like organelle, but they do share one important trait. They represent specialized regions in the cytosol that are devoted to a subset of cellular tasks. For example, eukaryotic cells contain 3 different types of fibers that serve as a scaffold for the organization of the cytosol, thereby earning them the name cytoskeleton. Careful arrangement of the cytoskeleton is essential for proper cell function. Without it, muscle cells would not contract, and nerve cells would fall silent. This video was created for educational purposes only. Codename Will, Professor Christopher Brunette, Leader of Research, Scientist, Ufologist, Psychic, Computer Programmer, Computer Technician, Philanthropist, Writer, Mathematician, Artist, Photographer, Film Producer, Chef, Animal, and Nature Lover. Passion, World Peace. Hope you enjoyed listening. Until next time, have an excellent day. This is a Professor, CBMD, PhD.